to trigger an early vegetative phase change in early flowering. Indeed, STTM 156-157-48 transgenic plants appeared delayed in leaf initiation and advanced in transition from juvenile to adult leaves following the production of the two abnormal, turbine blade-shaped cotyledons, Figure 8b. STTM 156-157-48 transgenic plants showed early production of adult leaves that were characterized by marked serration and trichomes on both the abaxial and adaxial surfaces, Figure 8b. Flowering was accelerated in STTM 156-157-48 plants as they bolted with 6 to 7 leaves, while the empty vector control plants bolted after making over 10 true leaves, Figure 8b. To determine whether STTM 156-157-48 similarly induced the reduction of Mir 156-157 levels, we performed Myrna gel blotting to determine Mir 156-157 levels. As expected, the abundance of Mir 156-157 was reduced by 90% in the STTM 156-157-48 transgenic plants compared with the empty vector control plants, Figure 8e. STTM is effective in triggering the reduction of Myrnas and Sirnas. A diagram of STTMs containing Mir 156-157, Mir 160, or tacrNAs, D7+, and D8+, binding sites. Orange indicates the spacer region. Blue indicates the bulge sequences in the Myrna binding sites. Red indicates the nucleotides that are different between the two Myrnas or the two tacrNAs. B, 2, D. Phenotypes of STTM 156-157, STTM 160-160, and STTM D7+, D8+, transgenic plants. E. RNA gel blotting to determine the levels of Mir 156-157, Mir 160, or tacrNAs, D7+, and D8+, in STTM 156-157, STTM 160-160, or STTM D7+, D8+, transgenic plants. Total RNAs were prepared from multiple primary transformants. We next used the STTM48 structure to target Mir 160, which negatively regulates auxin response factor genes, ARF10, 16, and 17, and plays important roles in many stages of plant growth and development. The STTM 160-160-48 transgenic plants exhibited hyponastic and serrated leaves, Figure 8c. These phenotypes were also observed in transgenic plants harboring Mir 160-resistant ARF10, Liu et al., 2007. Compared with MIM 160, STTM 160-160-48 transgenic plants displayed deeper leaf serrations, and the derepression of the Mir 160 target gene, ARF17, was also higher. See supplemental Figure 8 online, suggesting that STTM 160-160-48 was more effective than MIM 160. As expected, Mir 160 was decreased to an undetectable level in these transgenic plants, Figure 8e. To assess the effectiveness of STTM against endogenous Sirnas, we sought to target the D7+, and D8+, tacrNAs from Tacrna precursor 3, TAS3, using STTM, Figure 8a. D7+, and D8+, are different only in 3 nucleotides at their 3' ends, and both are generated through the Tacrna pathway composed of ARGONAUTE7, AGO7, RNA-dependent RNA-POLYMERASE6, RDR6, suppressor of gene SILENCING3, SGS3, and dicer LIKE4, DCL4. D7+, and D8+, target ARF2, 3, and 4 mRNAs to regulate developmental timing and patterning. Mutations in Tacrna pathway components abrogate the production of D7+, and D8+, and result in precocious vegetative phase transitions such that juvenile leaves adopt characteristics of adult leaves, including a higher length-width ratio. Indeed, the leaf morphology of STTM D7+, D8+, -48 transgenic plants resembled that of the RDR6 to 15 mutant lacking the TAS3 tacrNAs, Figure 8d. Small RNA gel blotting analysis revealed that D7+, and D8+, accumulated to a lower level compared with the empty vector control plants, figure 8e. Degradation of small RNAs by SDNs contributes to the reduction of small RNAs triggered by STTM. The SDN family of exonucleases was found to turn over small RNAs in vivo. Ramachandran and Chen, 2008. To test if the reduction of small RNAs triggered by STTM may be attributed to the degradation of small RNAs by SDNs, we sought in vivo genetic evidence in Arabidopsis. We generated STTM 165-166-48 transgenic plants in the background of the SDN1 to 1 SDN2 to 1 double mutant. The SDN1 to 1 SDN2 to 1 double mutant exhibited no obvious morphological phenotypes. Ramachandran and Chen, 2008. The great majority, approximately 95%, of the STTM 165-166-48 transgenic plants in the SDN1-SDN2 mutant background showed no phenotypes, and a small portion, approximately 5%, showed mild phenotypic alterations in the cotyledons at an early developmental stage, whereas at later stages the plants developed normally, figure 9a. By contrast, STTM 165-166-48 transgenic plants in the wild-type background showed visible phenotypes at a high frequency, approximately 90%. We analyzed the levels of STTM 165-166-48 transcripts and MIR 165-166 from the STTM 165-166-48 transgenic plants in the wild-type and SDN1 to 1 SDN2 to 1 backgrounds. Although the STTM 165-166-48 transcripts were only slightly lower in the SDN1 to 1 SDN2 to 1 background than in the wild-type background, figure 9b, the levels of MIR 165-166 differed significantly between these two genotypes. MIR 165-166 accumulated to higher levels in the transgenic plants in the SDN1 to 1 SDN2 to 1 background, figure 9b. To exclude the possibility that the slightly lower level of STTM 165-166-48 transcripts in SDN1 to 1 SDN the 2nd to the 1st of May compromise the degradation of MIR 165-166 in the mutant plants, we identified two independent STTM 165-166-48 transgenic plants in the SDN1 to 1 SDN2 to 1 background, in which the levels of STTM 165-166-48 transcripts were comparable to those of two independent STTM 165-166-48 transgenic plants in the wild-type background, figure 9c. Using quantitative RT-PCR, QRT-PCR, we found that although the two kinds of transgenic plants had similar levels of STTM 165-166-48 transcripts, the STTM 165-166-48 transgenic plants in the SDN1 to 1 SDN2 to 1 background accumulated a higher level of MIR 166 than that of STTM 165-166-48 transgenic plants in the wild-type background, figure 9d. The Darrepressed expression of MIR 166 caused by SDN mutations in the STTM 165-166-48 transgenic plants in SDN1 to 1 SDN2 to 1 background might account for the weakened phenotype. In summary, we found that mutations in SDN1 and SDN2 compromise the degradation of MIR 165-166 triggered by STTM 165-166. Degradation of MIR 165-166 by the SDNs contributes to the reduction of MIR 165-166 triggered by STTM in Arabidopsis. A. Phenotypes of vector and STTM 165-166-48 transgenic plants in the SDN1 to 1 SDN2 to 1 background, at right, compared with those in the wild-type, WT, background, at left. B. RNA gel blotting to determine the levels of STTM 165-166-48 RNA, MIR 165-166, and the U6 control RNA in the wild-type, SDN1, SDN2+, or SDN1 to 1 SDN2 to 1, SDN1, SDN2-, background. The numbers indicate the relative levels of the RNAs. C. Comparison of STTM expression in STTM 165-166-48 transgenic plants in SDN1 to 1 SDN2 to 1 background and in wild-type background. Bars show say. D. Comparison of MIR 166 levels in STTM 165-166-48 transgenic plants in SDN1 to 1 SDN2 to 1 background and in wild-type background. Bars show say. C. And D. Different shades of gray are used to indicate different independent lines to allow for the easy comparison between STTM and MIR 165-166 levels in the same lines. See online article for color version of this figure. Similarly, we generated STTM 156-157-48 transgenic plants in the SDN1 to 1 SDN2 to 1 background. We found that the proportion of abnormal transgenic plants was reduced from 30% in the wild type to 3.5% in the SDN1 to 1 SDN2 to 1 background. Based on the above observations, we conclude that STTM triggered target mRNA reduction is at least partially through SDNs. General rules for STTM design and application. Based on this study, we recommend the following general rules for STTM design. First, the STTM should be designed to have two non-cleavable mRNA binding sites linked by a spacer. The two non-cleavable mRNA binding sites can be either identical, to target one specific mRNA, or slightly different to target two slightly different mRNA members of the same family. To reach a maximal effect, we do not recommend using the current STTM structure to target two distinct mRNAs from different families due to insufficient degradation of mRNAs by STTM with only one mRNA binding site. Second, the mRNA binding site should have a CTA trinucleotide bulge corresponding to positions 10 and 11 from the 5' end of the mature mRNAs, so that the STTM can effectively bind but will not be cleaved by the mRNAs. Some small RNAs may coincidentally have the sequence tag after the 10th nucleotide, which complements the introduced CTA in the STTM. This may result in a shift of the position of the introduced bulge and a subsequent cleavage of the STTM. In these rare cases, a different trinucleotide bulge should be used instead to prevent the cleavage of the STTM. Third, the optimal length of the spacer is from 48 to 88 nucleotides. The sequences of the spacer should be relatively at-rich and able to form a stable stem, such as those used in this study. STTM is an effective approach for functional genomics of small RNAs. In this study, we explored the use of SNCRN STTM to block the functions of endogenous mRNAs and siRNAs, two major types of small RNAs in plants. Our results show that STTM is highly effective and specific in reducing the levels of targeted small RNAs in Arabidopsis. As such, this approach can be employed to explore the functions of small RNAs in planta. Although we only examined the effects of STTM in Arabidopsis in this study, STTM is also effective in other tested plant species, such as tobacco, Nicotiana tabacum, and rice, Oryza sativa. For plant species that are recalcitrant to transformation, it is possible that STTM